everyone. Welcome to today's webinars, Biosimilars and Their Use in Cancer Treatment. I'm Kate Fitzer, the Metastatic Breast Cancer Program Director at SHARE.
And I'm Jill Mancuso, a breast cancer advocate.
Before the presentation begins, I'd like to tell you a little bit about SHARE. We're a national nonprofit that supports, educates, and empowers anyone diagnosed with breast or gynecological cancers. We provide outreach to the general public about signs and symptoms, because no one should have to face breast, ovarian, uterine, cervical, or metastatic breast cancer alone. For more information about upcoming webinars, support groups, podcasts, and our helplines, please visit our website at sharecancersupport.org. Now I'd like to hand it over to Dr. Gralow to introduce herself. The screen is yours.
Thank you very much. My name is Julie Gralow. I'm a breast medical oncologist, and about 3 years ago, I joined the American Society of Clinical Oncology as the Chief Medical Officer. I also have had extensive experience in clinical trials with the USNCI's SWOG Clinical Trials Network, and I was the executive officer for breast and lung cancer prior to joining ASCO. About 30 years ago now, as I was just starting off on faculty, I was the co-founder of a group in the Seattle area called Team Survivor Northwest, which is a nonprofit aimed at helping female cancer survivors improve their health, their physical and their emotional health through fitness and exercise. And then I was also a professor of global health, and in that role formed the Women's Empowerment Cancer Advocacy Network, or WECAN, where I helped promote women cancer patient advocates starting in Eastern Europe, Central Asia, mostly the former Soviet Union in the beginning, and then in East and Southern Africa. So that's my background, and I am going to talk to you today about biosimilars. And do you want me to share my screen, or?
Yes, Dr. Green.
There we go. Okay, I'm going to get rid of some of these floating things. Okay, and I hope this is sharing okay. Kate, can you confirm if it's perfect? Okay, great. So thanks to SHARE and the team for inviting me to speak to you today on biosimilars and their use in cancer treatment. So let's start with what are biosimilars. It's kind of a funny name, and it's a little bit complicated. So biosimilar drugs are what we call biological products or biologics. So there are actually a lot of biologics in cancer treatment and beyond out there. And they're different from kind of a classic medicine in that they're generally very large molecules, and they're made from living sources, so they're grown up in bacteria, yeast, or animal cells. This is how you produce them. And monoclonal antibodies, there are a lot of antibodies now in cancer treatment. They're produced in cells, they're grown in big vats, if you will, and then you purify out the protein, these monoclonal antibodies. And so that's what makes it different from a classic molecule where you can just kind of, make it in the lab, and it's not grown in big vats. You don't have to purify it out. So vaccines are biologics. These monoclonal antibodies that I'll talk more about are biologics. They're very important in cancer. Insulin is a biologic, and then gene therapy. So biologics are used to treat a lot of diseases, including cancer. And I told you that they're usually big and complex and all, but insulin is considered a very simple biologic protein, whereas monoclonal antibodies are much more complex. And So where does this name biosimilar come from? Well, even within a brand name, biologic medicine, every time you make a big batch of it, you grow it up, the antibodies are made in these cells, you purify it out. The composition of that batch can vary a tiny bit in terms of the kinds of sugars you put on the protein, et cetera. So we say the composition of biologics can only be defined by a certain extent. Now, we have very narrow windows, and it's always tested. Each batch is tested to how much does it compare to the reference batch that was made early on. But there's a little bit of minor variability that happens, whether you're making the reference biologic, meaning the brand name approved drug, or whether you're making a biosimilar, which is a version of that biologic. And so as we make biologics, as we make biosimilars to those biologics, we have to be very carefully tracking the range of minor variability to make sure we're consistent in terms of safety and efficaciousness. So again, I told you a little bit about this. Here's how you make a monoclonal antibody. They are made from living organisms. And so you've got the cell line here, and you've got it's grown, in an incubator, and then you've got this upstream processing. You've got a big vat, then you purify it, you separate it, you characterize it, and then you put it in a vial. So again, I haven't stressed this enough. They're more complicated to purify, process, and manufacture than standard drugs. So biosimilars are biologics, and they have to be, by definition, highly similar to a biologic that's already FDA approved. So the originator drug, the one that we're copying, is called the originator or the reference product. I'm going to talk a bit about trastuzumab, a monoclonal antibody, the first one approved in breast cancer and the first one with a biosimilar. And the originator or reference product would be called Herceptin. Now, when I give talks, I'm actually not supposed to use brand names of drugs. That's how we're all taught. We're supposed to use the generic name. But in this case, it's really important to point out that the originator or the reference would be Herceptin, the one that was originally approved. In order to be approved as a biosimilar, the biosimilar can't have any meaningful difference from the reference or the originator product in terms of safety, purity, or potency. And by definition, biosimilars have to have the same route of administration. So if it's intravenous, it has to be given intravenously, the strength and the dosage form, and the potential side effects as the originator. So biosimilars are very similar, but not identical to the original biologic medication. And the FDA requires that there be no differences in the safety or effectiveness between biosimilars and the original biologic. They're made from the same sources. They give the same benefits when treating diseases, response rates, et cetera. Again, given at the same strength and dosage, and they're not expected to cause new or worsening side effects. Now, biosimilars have been developed with a couple of key goals. One is that they're more affordable. So just like much about generics, generics come on the market after a drug goes off patent, and now you have competition, and that usually drives down the price. But it's not just about the cost to insurance, to our government, to the patient with copays, et cetera. But it's also biosimilars were developed for equity purposes so that more patients could get access to these drugs because they're more affordable. And that's true within the United States, that lower cost drugs generally are more available. And it's also very true globally, that a lot of biologics are not put on the World Health Organization's essential medicines list are covered as part of a national cancer control plan solely because of the cost, not because of the effectiveness. So biosimilars can provide patients with more treatment options, increase access to the medications, and potentially lower healthcare costs through competition. And I've mentioned generics a little bit, but are biosimilars the same as generics? Why don't we just call them generics? I think I've given you the ammunition. You know, they are very similar, but they're not identical. And a generic is basically identical. So biosimilars and generic drugs are versions of a brand name drug. And the difference is the active ingredients of generics are smaller, simpler. They can be copied. Biologics can't be copied exactly because they contain a mix of many slight variations. And again, just to bring home the point, the generic is a term we use to describe a non-biologic copy. So they're smaller, less complex. They really are identical to the reference or originator product. Biosimilars, larger, more complex, come from living organisms, highly similar, but never actually exactly the same. So here's the timelines in Europe on the left and in the US on the right of oncology biosimilar development. So biologics are kind of a relatively new category of drugs, certainly in oncology. And as the originator biologic patents approached expiration, which meant that you could bring in competition, we had to develop what we call regulatory approval frameworks. So the FDA, the EMA, the European Medical Association, had to figure out how we're going to assess and approve biosimilars. In Europe, they started before us, in part because their patents generally wore off before ours. So Europe was ahead of us in terms of establishing a framework for how biosimilars would be evaluated at the regulatory level. In Europe, they approved the first oncology supportive care biosimilar, epoetin, erythropoietin. It's a red blood cell growth factor to keep your red blood cells up. And they approved their first therapeutic oncology biosimilars, trastuzumab, which is Herceptin, and bevacizumab, which is Avastin, in 2017. In the US, the FDA took longer to figure out our regulatory framework, essentially. And in 2010, we had something that came along as part of the Affordable Care Act called the US Biologics Price Competition and Innovation Act. In 2015, we approved our first oncology supportive care biosimilar, which was filgrastim or GCSF or Neupogen. That keeps your white cells up. Many of you may have been on that drug. And in 2017, the FDA approved our first oncology treatment biosimilars. Again, the same ones that they approved in Europe in 2017, trastuzumab and bevacizumab. So they haven't even really, the treatment biosimilars in cancer have not even been around for 10 years yet. But it's really speeding up. As of August, as of this month, there are 59 FDA-approved biosimilars, but they're only for 15 different reference biologics, and this goes across all disease types, not just cancer. 26 of them are oncology, but again, 59 sounds like a lot, but most of them, you know, the originator has 3 to 5 copies. So right now, for the cancer treatments or the cancer therapeutics in the US, we have 3 biosimilars. Bevacizumab, there are 5 different bevacizumabs to biosimilars. Trastuzumab or Herceptin, we have 6 different biosimilars approved. And for rituximab or Rituxan, which is used to treat lymphoma mostly, we have 3 biosimilars. For cancer supportive care drugs, we have 4 reference or originator products. I told you epoetin alpha, that was the first one that was approved in Europe. there's only one biosimilar version of that. Filgrastim, GCSF, Neupogen, there are 4 versions of biosimilars. Pegfilgrastim, which is Neulasta, that's a long-acting GCSF, there are 6 biosimilars. And just earlier this year, denosumab got approved as a biosimilar. I think it hasn't come on the market yet, or it's just coming on the market. And that would be Xgeva, which we use for patients with bone metastases to try to quiet down the bone metastases and prevent development of more. Also, in a different dosage and strength, we can use denosumab to prevent or treat osteoporosis. So I know you don't need to know or want to know probably all the details of how we develop these drugs, but for a brand new biologic, say we have a new antibody that looks like it targets something on ovarian cancer, the FDA has a clear pathway for the development of that biologic. And the bulk of the data, you start with a little bit of analysis and non-clinical work that might be in animals, and then you do a lot of analysis. But to get approved, you need big, huge clinical trials comparing the biologic to the standard of care to show that it's safe and effective. So the original biologic lays all the groundwork for targeting maybe that receptor on ovarian cancer actually works and it's not harmful. So there's an emphasis on safety, purity, potency. When it comes time to develop the biosimilar from that biologic, you've already proven that it's safe to have an antibody that binds to that target, for example, and that it doesn't cause excess toxicity. Everything causes some toxicity, but doesn't cause excess toxicity compared to the biologic. And then the clinical trials that have been mandated are much smaller because you're really trying to show that within a given window that they look like they're giving the same side effects and the same response rates and benefits as the originator. So biosimilar development, you don't need the huge trials showing that hitting that target with this antibody works. You need to show that it's similar to the reference product. And I know what concerns a lot of physicians and lots of patients is, how do we know that biosimilars are the same quality as the reference product? And the FDA outlines, how they're helping ensure us, the prescribers and the patients getting the drugs, that biosimilars are good quality. So they mandate a lot of research by the manufacturer to demonstrate high similarity. And then they do studies to show that there are no meaningful differences in safety, purity, or potency. And then they review all of that data. The FDA goes in and inspects the manufacturing facilities. And importantly, even after the biosimilar is approved, just like with any other drug, they do post-market, meaning after approval, drug safety surveillance that's required to be reported to the FDA, and the manufacturers have to track it as well. So biosimilar naming is kind of funny, and it's very confusing. So I told you already that, for example, for trastuzumab, we have 5 or 6 different biosimilars that are approved, and yet they all start with trastuzumab. That's the name of the antibody. And we need to be able to distinguish the biosimilars from the reference product, but the biosimilars from each other as well, so that if there were a problem, that we would know which one was causing the problem, right? So what the FDA decided to do was, you take the name of the core drug, which we're talking about trastuzumab, so that'd be trastuzumab, and they give 4 lowercase letters that are like randomly generated and put it after that. Now the companies can come up with their own trade names, and they do, but this is the generic nomenclature for the 6 different trastuzumab biosimilars that are out there and approved right now. So let's delve a little bit more deep into trastuzumab. It targets HER2, the human epidermal growth factor receptor 2 protein, which is found on maybe 20% of breast cancer. It is a monoclonal antibody. It's, we make our own antibodies all the time with our immune system to fight infection and things. And it was created in the lab. And when it's made, it's produced in Chinese hamster ovary cells. The reference biologic Herceptin, that was the first biologic approved in breast cancer. It was approved in 1998 in metastatic breast cancer, and then 2006 in early stage breast cancer. And then the first trastuzumab biosimilar was FDA approved in 2017, and it didn't launch. We didn't actually have anybody receiving these trastuzumab biosimilars until 2019. So it's only been about 5 years. And the delay between FDA approval and the US launch was due to patents expiring, settlement agreements between the manufacturers. There's lots of stuff that goes on where, the company that's been making the drug Herceptin for so long is looking to keep its patent life as long as possible. And yet, because of all this difference in the way that the US grants patent prolongation and protections, actually trastuzumab went off patent in India, and the trastuzumab biosimilar was available in India in 2014. So in India, we've got more than 10 years of experience using trastuzumab biosimilars. This is a paper that came out around the time that the trastuzumab biosimilars were being considered. And this is just showing the global need for a biosimilar because patients were not getting access to this really important HER2 targeted antibody. And so this paper looks over a 15-year period between 2000, when trastuzumab was first approved, I think I told you in 1998, for metastatic breast cancer. So this looks between 2000 and 2015 at the percentage of patients who had HER2 positive metastatic breast cancer who didn't get trastuzumab or any HER2 targeted agent. In the US, it was 12%, which means 88% were, but still 12% in the US did not have access to it for whatever reason. In Europe, the number who didn't have access to HER2 antibodies was higher. It ranged by country 27 to 54%. In China, it was about 27 up to 50%. And then trastuzumab was approved in early stage breast cancer later. So this period looked at 2005 to 2015 when trastuzumab was approved in early stage HER2 positive breast cancer. And people who still weren't getting it in that time frame with early stage breast cancer ranged from about 20 to 60% as you looked across the higher income countries, North America, Europe, Australia, New Zealand, China. But there was less than 10% availability of anti-HER2 therapies during these time periods in low and middle income countries. Real 90% plus of patients in rest of world and low and middle income countries were not able to get HER2 targeted antibodies, primarily because of the cost. So I've gone over what the general FDA strategy is for biosimilar. The trastuzumab biosimilars, they had to show that the antibodies that they made were highly similar in structure and function and binding to HER2. They then did clinical pharmacology, where you actually give these drugs. It could even be to healthy humans, but to humans in general. And they show that the way the drug is distributed and metabolized and excreted and the biologic response is the same as the originator. And then, especially with antibodies, we need to make sure that we don't get what we call immunogenicity, that the antibodies aren't causing us to react to these foreign antibodies. So we've got clinical trials that were done. that showed that the biosimilars were similar to the originator. And then we've got very active ongoing post-approval activities to make sure that they're safe and they're effective. And this is, I just showed you data from one trial. This was the Heritage trial that showed that one of the biosimilars to trastuzumab was compared to the originator, Herceptin, and they were given with a taxane drug. And relatively small study, about 500 patients. They showed that the response rate was similar, not statistically different. If anything, numerically, it might have been even a little better for the biosimilar, but it was statistically the same, about the same response rate. And the number of patients with one or more serious side effects was statistically the same. Now, I told you that biosimilars are approved at the same dose and the same route of administration as the originator. So very shortly after the trastuzumab biosimilars came on the market, now we had approval in the US for giving trastuzumab Herceptin underneath the skin as opposed to intravenously. And this just shows a picture of a subcutaneous injection. And so that means that now we can't give the biosimilar subcutaneously. That was an intentional strategy of the company that made trastuzumab, that they now got a new patent for taking trastuzumab and adding something called hyaluronidase that helps break down the areas so that the trastuzumab can go through the skin and get absorbed into the blood vessels. And they got a new patent on it in 2019. So we're not going to have subcutaneous biosimilars to trastuzumab or other antibodies until those patents expire. The patent for subcutaneous trastuzumab is due to expire in the US in 2030. So there are things that happen, at a market level to try to keep the branded name on the market for as long as possible. And this is one strategy. So right now, pretty much any antibody actually could be given under the skin, but they were developed to be IV infusions. And so this has been a strategy to prolong the patent. So this is what happens after approval. There's what we call pharmacovigilance. We're looking for product quality after approval. We look at long-term safety. Now, with biosimilars, and this might be why I've been asked to talk with you about it, is, there's both patients, but also physicians, have been slow to accept biosimilars. There's been, we call it maybe a nocebo effect, that if you know you're getting the non-branded biosimilar, you might, as the physician, report worse symptoms, as a patient report worse symptoms than if you thought you were getting the branded drug. There's been some concern about that. And then I'm going to go into a little detail about something called biosimilar interchangeability and substitution, because the European regulatory group and the US FDA have somewhat different positions. And I'll explain what that means in a little bit. ASCO, the American Society of Clinical Oncology, which is the professional organization I'm the chief medical officer for, we've really been reviewing this. Our job is to, help our members understand these new drugs, for example, understand issues, how we should use them, et cetera. So we've issued 3 different statements over the years on biosimilars and when you can change interchangeability, et cetera. And also, we issue clinical practice guidelines, and we really use evidence to try to make recommendations for what the best way to treat a given situation is. And so we've looked in our guidelines where we've recommended biologics of any kind across any cancer type. And for the most part, we've said that if we've said you can use trastuzumab or bevacizumab or this antibody drug conjugate, and if there's an approved biosimilar to that drug, as long as it's approved for the indication where we said you should give it, then we're saying it's okay to substitute. But we also said that the reflective switch between like the brand name, reference product, and the biosimilar without the knowledge of the prescriber or the patient is not recommended. So if there's a switch done, because now all of a sudden your insurance says you can only get this biosimilar and not the reference, or if your pharmacy is only stocking one version of the drug, if that happens, they at least need to tell you about it, both the prescriber as well, who then should alert the patient. And and so this whole issue of switching between the originator or the reference product and biosimilars has been part of the reason why there's been some slow acceptance, actually. And as I've already indicated, there could be a lot of reasons why treatment could be changed from the reference product to a biosimilar, from one biosimilar to another, or even back again from a biosimilar back to the originator product. But switching is typically done to decrease costs, make sure it's covered by insurance, or because of drug availability. You were getting one version of a biosimilar, and now it was cheaper to, for your pharmacy to buy a different biosimilar, and so they've switched. There is really strong real-world evidence that switching from a reference product to a biosimilar doesn't reduce treatment effectiveness or increase risk of adverse events. In Europe, they've allowed the switching, they call that interchangeability, without even letting anybody know which you're getting. They've been allowing it. So we've got lots of experience in Europe, for example, of switches that go back and forth without any negative effect observed on patients. And the FDA just did a big, what we call a meta-analysis of the clinical trials that had a switching and a non-switching arm between biosimilars and their reference products. And that big FDA analysis of the long-term results of many of these clinical trials of biologics and their biosimilars has not shown a difference in outcomes between the group that did switch and the group that didn't. So this is the study. It's kind of published on the FDA website of the FDA review of switching or not. It was just done in 2023. They were looking to see if it was safe to do in terms of toxicity and in terms of effectiveness. They looked at 31 studies that evaluated 21 different biosimilars to 8 different originator or reference products, over 10,000 participants across these studies. And they found there were no differences in the number of deaths, serious adverse events, or steady discontinuations due to an adverse event between those who were in switching arms of the study and those who weren't. And the results didn't seem to be affected by the product class or the direction of the switch or the number of switches. So that gets to what we call interchangeability. So switching and interchangeability. So in the US, we took a different strategy than Europe, and we made a regulatory pathway for approval of biosimilars. And then we added on top of that, you could do some extra tests and get your biosimilar named an interchangeable biosimilar. So you have to do everything for an interchangeable biosimilar as you do for a standard biosimilar, plus some additional studies, which are mainly switching studies. And the interchangeable designation in the US meant that the products could be substituted at the pharmacy level without the intervention of the prescribing healthcare provider. So if I wrote for Herceptin, the pharmacy could switch it with one of these trastuzumab biosimilars. Now, we do that all the time with generics. I don't know. I'm sure lots of you have pills that are filled all the time, and you get it and you you get the generic name for it. You're not sure which generic version of the drug you're getting. You're probably not getting the brand name much with pills anymore. So that's done all the time with generics, but with biologics, you have to have this interchangeable designation for the pharmacy to be the one who decides to switch. Now, it varies by state, and different states have different laws regarding interchangeable products, regarding the length of time to notify the prescriber. So basically, you do need to notify the person who wrote the prescription, but they don't need to rewrite the prescription, and how much time and advance notice you need to give to the prescriber varies state by state. Whether patients need to be informed varies state by state. And then how long the pharmacists need to retain their records about substitution varies from state to state. In Europe, the EMA considers all biosimilars interchangeable. In the US, very few biosimilars went all the way to get this interchangeable designation because it requires more time and money and work. None of the oncology biosimilars that I showed you have an interchangeable designation, but the FDA is revisiting this mainly based on that 2023 data, as well as the real world data that comes out of rest of world, showing that the switching doesn't seem to be harming patients in terms of disease response or control or side effects. I show you this if you're interested in learning more. This is the FDA's Biosimilars Action Plan that was developed in 2018. And just in April of this year, 2024, they actually published a summary of all the accomplishments. But the 4 key areas that the FDA outlined for their action plan back in 2018 were they wanted it to be efficient, not difficult, to get biosimilars approved. They wanted it to be clear. They wanted everybody to understand how you get this approval. They wanted to have good communication out to the public, to patients, to insurance companies, to clinicians on what biosimilars were, and they wanted to promote competition. So you can see their summary of what they accomplished over that 6-year period if you're interested. And this is something that just got posted in June of 2024 on that interchangeability, that the FDA is in the process of updating its guidance to the pharmaceutical industry on interchangeables. And I put here on the slide a quote from Dr. Yim, who's the director of the FDA Office of Therapeutic Biologics and Biosimilars. She says, "We've gained valuable experience reviewing both biosimilar and interchangeable biosimilar meds in the past 10 years. Both of them meet the same high standard of biosimilarity for FDA approval. Both are safe and effective as the reference product. And the recommendations in today's draft guidance, when finalized, will provide clarity and transparency about the FDA's thinking and align the review and approval process with existing and emerging science." So essentially, if you read between the lines, they're thinking of taking away the need to do these extra studies for interchangeability. Now, this is up and posted. It's draft guidance, and it's up for responses and comments on it at the present time. And this kind of comes out of their statement. It's a misconception that because an interchangeable biosimilar designation meets additional requirements, that it's safer. Experience shows that biosimilars and interchangeable biosimilars have the same safety and effectiveness. And the FDA says that because some biosimilars are now interchangeable and others aren't, that it actually creates the illusion that it's easier to switch if you've got this biosimilar interchangeability designation when it's not actually the case. And they're basically saying that with all the experience, with all the biosimilars approved to date, there's really no risk between switches. So here's what's happened since biosimilars had a pathway for approval in the US. It's estimated that the cost savings to the United States, whether it be to the payer, the government, the patient, for using biosimilars between 2015 and 2019 was $19 billion. And that the projected cost savings that'll occur in the past 4 years, 2020 to 2024, is 104 billion US dollars. That's only in the US. And this comes from a report of the Association for Accessible Medicines, if you want to read it. So biosimilar savings just is exponentially increasing. It's been increasing, the savings has been increasing by at least 2 billion per year. So each year the savings is $2 billion higher than the year before. And what's leading the charge are the 3 oncology treatment biosimilars: bevacizumab, rituximab, and trastuzumab. They are saving the most money. Biosimilars have had a positive impact on health equity. This comes from that same report from the Association for Accessible Medicines. They are delivering on their promise of expanded patient access. Of the 700 million days of therapy that US patients have had with biosimilars in this report up to 2023, 344 million days they estimated to be days that patients got these drugs that they otherwise might not have received because of coverage issues, cost issues, et cetera. And biosimilars are helping improve access to medicines that wouldn't have normally been covered or would have been unaffordable. So biosimilars do face challenges to adoption and sustainability. Although it's improving, the adoption of the use of biosimilars in the US in particular has been slow. 2 of the biosimilars out there have adoption rates of greater than 80%, but others are as low as only 13% of the time a prescription is written for that drug as a biosimilar use. There still are misconceptions, lack of education awareness among physicians and patients, and in industry and in practice and in the payers, we have misaligned incentives that are slowing it down. In some ways, there are incentives that exist in our system that the more a drug costs, the more you get paid or get rebates on or whatever. So we've got some incentive issues that are misaligned with the adoption of biosimilars. We have 106 different biosimilars that are currently in development. I'm sure that number is wrong already because there's more on the market that are being developed all the time. But there are a lot that aren't being developed. So 86% of the brand name biologics that would be eligible to create biosimilars don't have a biosimilar under development right now. And the high cost of development and the uncertainty regarding the future of the biosimilar market in the US is a bit of a problem. And that's what's causing many potential biosimilar manufacturers to not even develop a biosimilar for a drug. So they'll be ready to go at the time that drug goes off patent. Here's a really good reference for you if you want more information on biosimilars. It was just updated in June from the US FDA, 9 things to know about biosimilars and interchangeable biosimilars. So in conclusion, biosimilars can reduce treatment costs and improve patient access to biologic therapies without evidence that they compromise quality of care. The extent to which biosimilars are being integrated into cancer treatment is depending on the acceptance by those who are prescribing, the pharmacists, the regulators, and patients. And we absolutely have opportunities that remain for improved awareness, acceptance, education, and knowledge. And for that reason, since we have opportunities that remain in the area, I really thank Cher for inviting me to give this talk on biosimilars and to offer the opportunity for you to ask your questions about them. And I'm going to stop screen sharing. There we go.
Thank you for the excellent presentation, Dr. Gralow, on biosimilars. Now we're going to start with the Q&A. You can still submit questions in the Q&A section at the bottom of your screen. We will try to get through all of the submitted questions, but may not be able to do so due to time constraints. So I'm going to start with the first question. Somebody is asking, are there any biosimilars being used to treat ovarian cancer?
So one of the three therapeutic biosimilars, bevacizumab or Avastin, is sometimes used in the treatment of ovarian cancer. We have other biologics being used across the gynecologic cancers. For example, we have these immune checkpoint inhibitors, immunotherapies you may have heard about. They're antibodies as well. They're all still on patent, but they are biologics and they will go off patent. There's a very cool new antibody drug conjugate. So I didn't even talk about those. We've got those for triple negative breast cancer, as well as HER2 positive breast cancer, a lot of other cancer types. But an antibody drug conjugate is taking one of those antibodies and then linking it directly to a drug. So the antibody brings the drug inside the cancer cell. The link between the drug and the antibody is cut off, and now you've got chemo delivered inside the cancer cell. So there's a new antibody drug conjugate that's been approved in ovarian cancer. Now it's going to have a long patent life. This is a fairly new drug, but it's just an example of another ovarian cancer drug that's a biologic that someday will be available, hopefully, in ovarian cancer.
We have a live question that we'd like to have answered. Herceptin has many biosimilars. Since they must all be different yet very similar, how do the drug companies maintain the effectiveness while being different from each other? And what is the change in that formulation to make them different so another company can produce it?
Yeah, so by design, the antibodies, the basic structure of the drug, are essentially the same in all the key points, the points that bind to the HER2 molecule in the cancer cell, the part at the end of the antibody that brings in complement and the T cells and everything else. There's just little minor differences in areas that don't really, if you will, matter. There might be minor differences in how the sugars are put, the body puts sugars on it. Now, what's very interesting in that question is when trastuzumab came out, one of the first antibodies that we used for cancer, even within the originator, Herceptin, the company is obligated to look at each batch and make sure it's not drifting. And there was some early evidence that when Herceptin was being made, they saw this drift in terms of the molecule just changing a little bit from batch to batch. And they had to go back. I don't know how many times they've had to go back, but when they saw the drift, they went back to an original cell line that was frozen down and then kind of brought it up. So even not in biosimilars, even just biologics more broadly, you can get a little bit of drift, but you're monitoring it and you can bring it back and go back to where you started from. So the answer to the question is, what's different about the 6 trastuzumab biosimilars? They really are an antibody to HER2 that, at least for the binding parts of it, the key parts of it, is identical. There's not much to distinguish any of them, if anything.
Thank you.
Okay, we have a few questions about lobular breast cancer and whether biosimilars are equally effective for treating lobular breast cancer when compared with the ductal carcinoma or ductal breast cancer.
Yeah, good question. We're always looking at where can we pull out data on lobular breast cancer. So right now, very little lobular breast cancer is HER2 positive. And so the main biosimilar that we have for breast cancer is trastuzumab, which binds to HER2. So you're not going to have very many, lobulars, because lobular generally is positive for estrogen receptor and progesterone receptor and negative for HER2. Now, this is going to become relevant because we have an antibody drug conjugate now to HER2, but trastuzumab deruxtecan, that you've seen data that not only is it effective in classic HER2 positive breast cancer, meaning 3+ or FISH positive, but it's also effective in 2+, 1+, and even some HER2 0, where you just have a faint amount of HER2 on the cell. That's going to bring in the lobulars, right? If you can get down to these low levels of HER2 that we would have classically considered HER2 negative, then you've got the lobulars. So we will be getting data on how are the lobulars doing with this antibody drug conjugate that's basically trastuzumab plus chemo attached to it. But that won't be available as a biosimilar for a long time, if that makes sense.
So, how long after a patent expires that we are seeing the biosimilars being approved and available to patients?
Sometimes it's pretty immediate because the companies know when patents are being expired, right? So they're developing them. And also because rest of world generally has patents expire before the US because we are a little bit more lenient in patent prolongation, et cetera. You know, companies have geared up for rest of world before it goes off, US patent. So in a highly competitive market, trastuzumab, bevacizumab, rituximab, the 3 treatment biosimilars we have in oncology, those are commonly used. You know, there's a big market for that. So there was a lot of interest to have them ready to go and get them out there as soon as the patent expired and all the legal stuff was taken care of. But there are going to be other biologics, and I talked about how many potential biologics aren't being developed as biosimilars. And that's because the market is so much smaller. So while it's costly to develop a biosimilar, it's much more expensive than making a classic generic drug, you know, which is much easier to produce, et cetera. So that's why we're not having it developed for biologics where there's not a big market. So it kind of depends on how big the market is.
We've had several questions about hormone replacement therapy, or HRT, and bioidentical hormone therapy. Are bioidentical hormones the same as biologic or biosimilar drugs?
That's a good question. You'd think from the name, bioidentical, that they would be, but they really aren't. And because the biologic or a biosimilar, it's how the drug is manufactured. And in this case, with kind of the bioidentical use of estrogens, of hormones, you're just trying to match what's in the body. So that's where the bio part gets in, but they're not made, they're more generic, if you will, than so it's all about the way they're made. They're not made in a vat, you know, in living organisms. You're just trying to match them to what's in our own body. So they're not.
Thank you.
So you had mentioned that the efficacy is the same, right? Because we have studies to show that, and you talked about like adverse side effects. But I wonder because, you know, in the generic area, a lot of us have taken, you know, the aromatase inhibitors, and we do notice there's a difference between the different medications, and you can have different side effects. And while it might not be an adverse side effect, possibly there is a side effect. And there's so many biosimilars for each drug that you had mentioned. Are there side effect differences between all the biosimilars, and is it easy for a patient to switch? Is it okay to switch to see if there's a
Yeah.
difference?
So great question. With pills, and when you see differences between, you know, generics and the brand name, the difference is usually explained not by the active component of the drug, but by the filler, what it is that keeps the active component of the drug in the pill form. So it's actually the extraneous stuff that you're reacting to, not the chemical structure of the drug itself, because that's identical. So it's how they package it, basically, to keep it all together. With biosimilars, they're virtually all given intravenous at this point. And you don't have, could you have a little difference in the way they're packaged? You could. I'm not aware that they're adding other things, though, to keep them in solution, or different preservatives. But I suppose in theory, there could be maybe a slightly different preservative or something, but that would be what's accounting for it. It's not the drug itself. And then you said, can you ask if you think you're having a different reaction or something? Um, you know, of course you can ask, it's your body, it's your, but whether or not the, insurance will approve it unless you kind of got some good evidence, that is unclear, so it's making the case to the person who's paying for it, and that's where the barrier comes into play. I mean, but if you have, you collect your symptoms and you've got some good evidence, it's absolutely appropriate to ask. Of course it is.
Wondering, how do you address concerns from patients who might be hesitant to switch from a biologic to a biosimilar? Let's say if that were the case.
Yeah, well, that's why our uptake has not been as fast as in some other parts of the world. I mean, I think we have to have open communication, but we have to, try to show the evidence, uh, and hopefully reassure. It's complicated, but what's interesting to me is, and especially if you're doing really well and you're on a drug and you just don't want any disruption, right? I can absolutely understand why they might be concerned, even if the FDA is saying, hey, look, we see no difference, if you switch or not. So I think it takes a while to get comfortable with something like this. And I think, I have real concerns about if we, as it seems we are, kind of giving this interchangeability to all drugs, that you wouldn't be at least mandated to tell the person writing the prescription and the patient who's going to get the drug that is being switched. I mean, that's of a concern. And right now, there are some states, if you have an interchangeable, where you don't necessarily have to do that. So that's ASCO's stance is the prescriber should know. So not only do they have it in their records and they know, but they can notify the patient and the patient can be aware as well. It's a real conversation. I would say the majority of my patients, if I show them the data and the safety and what the FDA has done and how many more years other parts of the world have been using these drugs with long-term data, it's very reassuring. But sometimes you get very comfortable with it. When trastuzumab, the subcutaneous version came out, I thought a lot of my patients would want to switch. It's easier to just get a shot underneath the skin than to get your IV hooked up. But I had a good group of patients who said, I am just so comfortable with the way things are right now. I don't want to rock the boat at all. And that was okay.
Thank you. Very interesting. Thank you so much, Dr. Gralow, for your thoughtful and thorough answers to our many, many questions. Also, please make sure to check SHARE's website for upcoming educational programs, podcast episodes, and support groups. And don't forget to follow us on social media as well.
This concludes the webinar. Thank you again, Dr. Gralow, and I hope that everyone has a pleasant evening. Thank you.
Thank you.